That's how respiration works, as we'll see. And then after we go through all of respiration, we'll kind of drop back a bit and look again at fermentation. We'll see that fermentation is basically, it's just this guy. It's just glycolysis, but with a couple extra steps to get rid of some waste products. That's all it really is. It's kind of glycolysis by itself with a couple extra steps. All right? And then we'll also see how lipids and proteins are broken down, because we're mostly going to be starting by focusing on sugars. Yeah, the carb. All right? So I found this diagram on the internet when I was looking around enzyme stuff. This is a diagram of an active site of an enzyme. Okay? In the active site of an enzyme, there's a nucleotide here. You guys see the adenine, there's the ribose, and this phosphate is double linked. You guys might have actually heard of this before. This is, see how the phosphate is linked both to the 5' and the 3' carbon? It makes a cyclic structure, and it's one phosphate. So this is cyclic adenosine monophosphate, cyclic AMP. Heard of cyclic AMP before? Yeah, so this is the enzyme that produces cyclic AMP. And the reason it produces it so well is it binds it well, because you see here, there's a hydrogen bond donor working with this hydrogen bond acceptor on the phosphate. All right, there's a cationic amino group interacting with an anionic oxygen here. There's a hydrogen bond donor interacting with this oxygen of this hydroxyl. And look, the hydrogen of the hydroxyl is acting as a hydrogen bond donor to the glutamic acid oxygen right here. And then the hydrogens on the amino group on the adenine, they're hydrogen bonding to I can't remember which amino acid that is, but this little amino acid, and then that's not serine. What is that? I'm blanking. Anyway, and then we got hydrogen bonds going on here. So basically, you see how not only is the shape a perfect match for the substrate, but the interactions are perfect to hold it right in place. Okay. And it's not just holding it there kind of passively. There's a little bit of tension going on, right, in interacting with it. So these proteins are these big molecules, but the right place, and what do we call this place? The active site, to hold a substrate just right. So it'll line things up or start to kind of bend and twist and strain things so that they're ready to react.
I'm sorry, so the substrate here is cyclic AMP?
This is cyclic AMP, yeah. We'll learn more about that one later. All right. So last time we talked about physical factors that affect enzymes. We're gonna talk about chemical factors that affect enzymes now, and then we'll jump right into the metabolic pathways. So there are chemical factors that affect the activity of enzymes, and they can interact with enzymes in different ways. Two different ways, really. Two main different ways. So in this case, you have our substrate, and under normal circumstances, the substrate should interact and bind properly with the active site. Okay, so you guys see how the shape of the substrate, and it's going to have the right charges and polarities and things to just kind of fit right in that pocket, right? And then the enzyme's going to go, right, and just kind of relax around it. And that's when the chemistry starts to happen, and we break it into its products. But sometimes you can have another molecule that looks a lot like the substrate, or even a little bit like the substrate, or part of the substrate, and it might fit into the active site or part of the active site. And so it only partially fits in the active site, so the active site doesn't do anything to it. No chemistry happens. But it just sits there, and it kind of stays there. All right? Actually, just to clarify, I'm going to be a biochemist with you guys, because I am. When things bind, they don't just sit there. All right. Everything in chemistry, unless there's a covalent bond made, if there's just weak bonds, it's pretty much yeah. All right. They don't just sit there. They kind of release and come back. They probably spend most of their time in that position, but everything kind of diffuses away for a moment and then might come back. Right. So they're spending most of their time in that position, but not all their time. All right? And so because it's occupying part of the active site, what does that do for our substrate? Yeah, it can't bind. It's like boink. All right. All right, no help. Okay? how can we overcome this problem? Remember things. So let's put it this way. There's an equilibrium between binding and unbinding. Binding and unbinding. It's an equilibrium. Everything that goes towards equilibrium, right? Even binding and unbinding. So, as it says, because it takes up the active site, takes it away from the substrate, it's called a competitive inhibitor. Right? That's the definition of a competitive inhibitor. It binds to the active site and competes with the natural substrate. Okay? So if it's competitive, how can we outcompete it? More substrate. Yeah, because if there's more substrate, if there's a lower concentration of inhibitor than there is of substrate, then the substrate will basically can start to outcompete it, right? It can substrate will spend more time interacting with the active site than the competitor would, all right? Or if you want to shut it down, you do the opposite. Okay, you add more competitive inhibitor, then it gets outcompeted.
Is that like so basically just a different concentration of the
Yeah.
substrate? Okay, not
Higher
necessarily like size or anything.
No, it's concentration.
All concentration.
Yeah, it's concentration. Yeah, if it was a different size, it wouldn't fit. Right? Because that size and shape is very, very specific. So I've got a little video here, see if I can get it to work without killing my presentation, but it probably won't. Activated. Go. Well, I guess we'll have to come back to it. It says that website's not working. Sometimes I find these links and then like a few weeks later they don't work anymore. All right, so that's competitive inhibition. All right. What else was I going to say about competitive inhibition? Oh, so there's this enzyme in our bloodstream. You ready? There's this enzyme in our bloodstream, and it breaks down fatty acids in our blood and turns them into lipids called prostaglandins. And when prostaglandins are produced, prostaglandins interact with the walls of our blood vessels, and they basically stimulate inflammatory type responses. And you know what happens to your blood vessels when there's an inflammatory response? They dilate. Okay. So it can cause some vasodilation. Also, if it builds up enough, it can actually go outside the blood vessels and start affecting nerve endings. And when prostaglandins reach your sensory nerve endings, our body interprets that as pain. And when prostaglandins go to your brain, they have receptors in the hypothalamus, and the hypothalamus says, something is not quite right. Let's turn the temperature up a little bit. So if you have too many prostaglandins in your system, what do you get? Fever. Okay? So, but the same enzyme, it's called cyclooxygenase, the same enzyme that makes prostaglandins, because prostaglandins have multiple effects, it affects blood flow, it affects pain perception, and it affects fever, so body temperature. If you use a competitive inhibitor to inhibit the cyclooxygenase, then you get less fever, less pain, freer blood flow. Okay, less clotting. It actually contributes to clotting too. All right. Any idea what this competitive inhibitor is?
NSAID?
Hmm?
NSAID, like a Advil?
Yeah, basically aspirin is the original one. All right, and all the other ones. It's a competitive inhibitor of cyclooxygenase. All right, so it blocks binding of the fatty molecules that get synthesized into prostaglandins. All right, that's how it works. Competitive inhibitor. Where did I put my notes? There it is. All right. All right. Here's another example of a competitive inhibitor. Here's a drug called PABA for short. It's para-aminobenzoic acid. Has anyone taken organic chemistry? Yeah. All right. So this ring is called a benzene ring, and it has a carboxyl group, a carboxylic acid, so it's benzoic acid, okay? And then if you're at the opposite side of the ring, it's called the para position. So it's para-amino benzoic acid. Just giving you some fun organic chemistry naming, all right? But there's a drug that is synthesized that has this sulfoxide with an amino group I don't even know sure what you call that group exactly. It's a funky group. But basically, instead of a carboxy group, it's got this thing. And so it's got at least 2/3 of the structure of this molecule the same. And this binds to the enzyme that converts para-aminobenzoic acid into its final product in cells, which is folic acid. All right. Remember I mentioned folic acid to you guys before? Folic acid is a coenzyme, right? It's a vitamin for us, but bacteria make it themselves. But if you block the enzyme that makes folic acid, bacteria it's not a vitamin to them. They don't go out looking for it because they make it themselves usually. And so if you block it, they can't make folic acid for themselves. And do you remember what folic acid was for? Yes, lipid synthesis. Okay, they can't synthesize lipids, and if you can't synthesize lipids, cells don't grow because you can't make membranes, you can't make lots of stuff, right? We don't even have this enzyme, so that makes this a really good antibiotic. All right, and so these sulfur-containing antibiotics are called sulfa drugs. You might have heard of those. The only problem with them is that some of them are pretty allergenic. People develop allergies to them. But see how that works? But it's a competitive inhibitor of the enzyme. And I can't remember the name of the enzyme. I should look that up. All right. All right. Non-competitive inhibition. So non-competitive inhibition is going to introduce a concept, again, that we will see for the rest of the semester. I don't know why I'm carrying my goggles on. I just love my lab goggles. All right. So, non-competitive inhibition, nothing else is binding to the active site, so that's why we don't call it competitive, right? It's not competing with anything. But there's a separate site. See this little guy kind of hiding over here, being all nondescript and everything in the shadows? All right. So that site over there is called an allosteric site. Have you guys heard the word allosteric before? All right. This is a super important word because it explains a whole bunch of stuff in cellular biology and biochemistry. Basically, allostery is how pretty much all proteins are regulated. Just about all, right? There could be some competitive regulation, but allosteric usually is involved in something in almost every protein, whether it's a transporter protein, a membrane signal receptor protein, an enzyme, whatever it is. Allostery comes into play all the time. All right. So allosteric. Allo means different or a change. Right? Something is different or something has changed. You with me? All right. And steric, if you remember your chemistry well, steric means there was a change in shape or position or something, right? So something physically moved around, okay? So allosteric means
Change in shape.
change in shape or different shape, okay? And what did we learn? If you change shape, you change function, okay? So this non-competitive inhibitor, it binds over here. You guys see that? It can bind way, all the way across, North Dakota on the other side of the enzyme. But when it binds here, whenever something binds to a protein, what does a protein do? Changes shape. So what happens is it binds to that allosteric site, and the allosteric site, just like an active site, will kind of grip it. Right? It's a specific interaction. Everything is specific with proteins. All right. So it grips it, and these chains move. But when these chains move, that causes the chains next to them to also move, and the ones next to those move. And so all the chains are kind of moving all throughout the protein in sort of a wave, and it ultimately changes the shape of the active site. And if you change the shape of the active site, it no longer recognizes the substrate. Okay? Right? And this is actually, this method, well, competitive inhibition too, but this method is very commonly used in what is called feedback regulation of our metabolism. And so often some small molecule, often in the same pathway that this enzyme is doing its thing, if there's too much or too little of it, usually too much, it will bind and it'll shut off that enzyme. And it says, hey, we got enough, let's turn you down for a while. All right, so that's how our metabolic pathways are often internally regulated. Right? It's not like your cell has to go, oh, we've decided we should do this, and starts making new stuff to shut things down. That's not what it does. The pathway itself will often self-regulate. that make some sense? Okay. So that's how that works. So that's non-competitive inhibition, or it's also just called allosteric inhibition. All right. And you mostly hear about allosteric inhibition, but there is also allosteric activation. Right? They're both very common. So you could bind. You can have an allosteric activator, and so it could bind to the protein, and maybe the active site was in an inactive form, and it goes into its active form. All right. So you can also activate things that way. All right. So speaking of feedback inhibition, so there's feedback regulation, in this particular case, feedback inhibition. Remember we talked a little bit about biochemical pathways before. You have multiple steps, and multiple steps, each one's a separate reaction, and each separate reaction requires its own enzyme. Okay? So here's an illustration of sort of a generic biochemical pathway. So we have some substrate, and it binds to the first enzyme. Once they form an enzyme substrate complex, then it's going to convert the enzyme into the first product, which they call the intermediate, because it's not quite done yet, right? But it's the first product is what that is. And then this product 1 becomes the substrate for enzyme 2, and enzyme 2 makes its product, all right? Product 2, or product B, let's call it, and product B becomes the substrate for enzyme 3, and then apparently this one does a whole lot of stuff because it really changed. I was like, wow. Usually enzymes only do like a few little modifications at a time, but that's pretty serious. And so basically, this pathway is making something that the cell is using. Okay? If you come upon a situation where the cell no longer needs or is using this pathway will just continue because it doesn't know any better. Right? It'll continue doing that, and eventually the concentration of this product, it's not being used, it's not being taken away, so the concentration starts to build up. And if it builds up, it can bind to an allosteric site. Sometimes it will even compete at the active site. It can be, so feedback inhibition can be competitive. Right? You're like I told you guys, in biochemistry, the book kind of oversimplifies and misses some things. So just listen to me, I'll tell you, all right? I will light your way. And so it binds to the allosteric site, and that changes the shape of the active site in particular, and it can't bind substrate. But remember, even non-competitive, even allosteric inhibition, that's at equilibrium. So it's not like it's binding 100% tightly, right? It's not covalent. And so it kind of binds and unbinds, binds and unbinds. It just means it turns it way down, so that every once in a while the active site will pop into proper conformation, but most of the time it's not fitting that substrate well. Okay? Everything's at equilibrium. All right. The only time you lock something in place is if you actually put a covalent bond in there. That's not what our cells do naturally. The things that put covalent bonds on active sites or even allosteric sites in enzymes, we usually call those toxins, or at least very powerful drugs. Let's see if I can remember now. It just occurred to me that there's a nasty one. What's it called? Sarin? You guys know the nerve gas?
Sarin gas.
Yeah. Let's see if I can remember right. It is a permanent, so it's a permanent competitive inhibitor of, I believe, acetylcholinesterase. You guys know what acetylcholinesterase does? in your neuromuscular junctions, it breaks down acetylcholine, so after a muscle is stimulated and fires, it can be allowed to relax. All right? Yeah. So the body just seizes up and you stop breathing. Because you need skeletal muscles to breathe, all right? So whenever you think like, well, the person's paralyzed, but everything else should be okay. So no, because skeletal muscles help you breathe. Your heart will still function. A lot of autonomic things will still function, but not breathing. Yes.
So you said for competitive inhibitor that the substrates can be more, and then these can compete the.
Yeah, so if substrate starts building up,
Yeah.
then it can start to outcompete, and the activity will go up a little bit. But it will permanently be slower because there's still something competing.
What would be? Is there any competition for the allosteric site?
No, usually you just remove it. Yeah, you usually just, it stops being produced, or you stop taking the drug, or whatever it is. Yeah.
Does it ever inhibit its own pathway, or is it always inhibiting?
Oh, that is its pathway.
Oh, that's the same.
Yeah, it's still enzyme 1.
Oh, okay.
Yeah, so see, it shuts down enzyme 1, and so you guys see, why would you do that? All right, you're not using this anymore, hint. So it starts to build up. If you have excess of something, it means you don't really need it. Okay? And so another thing to think about, because I'm a biochemist, is the allosteric site has kind of a low affinity. So that means there has to be a lot of this around for it to really bind. Okay? It's not like, oh, there's a couple extras, we're going to shut down. It doesn't do that. It has to kind of build up for a little while, right, for it to actually shut down. It's got a fairly low affinity. The active site has a much higher affinity than the allosteric site does. You don't want it just shutting down randomly. It's like metabolism's going great. It's going great. You get little hiccups in your metabolism, right?
Oh, what about like positive feedback? Does that have the same site?
Positive feedback is a physiological thing. It's not really a biochemical level thing. Yeah, so this is feedback. So it's not, notice I didn't say negative feedback. It's feedback inhibition. There's a difference. This is a biochemical thing. That's a physiological thing. All right. Ooh, let's see if I can get this one to work. Come on, baby, you're good. Just thinking about it. Oop, it killed my presentation. Thank you, PowerPoint. Fix it just so. Alright, found it. To enable project there. Yes, just a moment. Are you guys ready? Where's the sound?
Cells, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction.
Product 2.
If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed.
So if you're not
In
using something,
feedback
you don't want to keep
inhibition,
making
the
it.
end product of the pathway reacts with the first
Cells need to be
enzyme
really,
that is
really
unique to the pathway.
Efficient, right? So
The
always think
reaction
about
occurs
that.
at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final
Rejected.
product is no longer synthesized.
We stopped making strawberry jelly bellies.
So it only attaches, it will only find an allosteric site on the initial enzyme because it wants to shut the process down from the very top, right? That,
Yeah, so usually you shut the process down as early as possible.
okay.
Okay, makes sense?
Mhm.
Because if you shut it down over here, then you make stuff and it just kind of backs up and you're wasting things. Okay, so you shut it down as early as possible. And when I say as possible, I mean the earliest step that's specific to that pathway. Remember we talked about the branch? And so stuff before here might, if you block this, then this won't happen. And then the precursor molecules can go off into other pathways, right? Because things basically just shunt, wherever the flow is. If you put a dam in this river, they just go down the other streams. Right? Yes.
Did you say this is how every enzyme is regulated?
Not every enzyme, but almost every protein has some. Well, I'll say most proteins have some kind of allosteric interaction with something in their function. I mean, even binding the substrate is almost like an allosteric thing, right? But usually it means a separate site. But technically, allosteric just means different too. All right, so we can open my presentation. Where were we? La, 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 la. Over here. All right, we did that one, did this one. amoeba sisters, enzyme and substrate. See how much they love each other? All right, so remember I told you guys, this is more like the lock and key theory, right? Where the substrate just fits right into the active site. But what's shown to be much more accurate is the induced fit, right? see how they just form a nice little hug. Okay, and they fit, it sort of conforms to the substrate, so they fit better, and it gives them a more specific, right? Because they contact so much, right? Remember, I showed you that diagram of all the different groups that interact with the substrate. It's very specific, right? It's not just oh, the shape's kind of right. That's only like the minor part of it. It's all the hydrogen bonding and hydrophobic interactions and ionic bonds and all that good stuff. All right. So the main reactions we'll often concern ourselves with during the rest of this chapter are good old oxidation reduction, which we call redox reactions. And why do we call them redox reactions? Well, they always happen together. If something gets reduced, something else must have gotten oxidized. And if something got oxidized, something else got reduced. Okay, one doesn't happen without the other. All right. So what is oxidation? Oxidation means that you're going to take away some electron. So here's a molecule. It's got like an extra electron, or at least an available electron, I guess I should say. So it's got an electron that's available, and B is going to take away that electron. All right. So B is what we call the oxidizing agent. All right. So it takes the electron away, and when it takes the electron away, molecule A becomes oxidized. And since B, the oxidizing agent, took the electron, B has become reduced. Wasn't that fun? Okay? They're just passing the ball back and forth. All right? So oxidation is the removal of electrons, right? Or loss of electrons. You guys know the mnemonic? Yeah, I didn't even know that until students taught me that a few years ago. I just knew oxidation, you take away electrons. Yeah, oil rig, oxidation is loss, reduction is gain, rig. All right, so reduction is gain of electrons, all right? So a redox reaction, those two are always paired together, right? It's a reciprocal process. It's like, if I lose an electron, you must have gained the electron, all right? Maybe I want it back. But you probably won't give it to me, so let's move on. Ooh, okay. All right, so here's a question. Just put it in your notebook because I didn't bring all the blue books. But in a redox reaction like the one here, which one carries more energy? The oxidized product or the reduced product? And explain yourself. I'll give you a minute to think about that and we'll talk about that. I don't. I said earlier I forgot to bring them down. So just write it in your notebook. I had them in my hand and somehow I didn't make it down here. Oh. That's Dr. Bahag's collection of pens down here. She started a new career this year. How many people went to the talk last night? Didn't you like how Professor Young put active learning into her actual presentation? Made you guys talk to each other. All right, go ahead and discuss it with your neighbors.
Yeah, I was just saying, she hasn't. That's curious. That is like, I and he was joking, I made a big reference to back
All right, who's got more energy? The oxidized or the reduced product? Reduced. Why would the reduced have extra energy? Because it has an electron. And electrons carry energy. Right? I have the power. No. All right. If you have the electrons, you can be the master of the universe. All right. How did the Infinity Gauntlet really work? It manipulates electrons. That's my hypothesis. All right. All right, so in biological systems, the electrons, all right? The electrons are what carries the energy. But electrons don't just like fly around by themselves in space, all right? That doesn't really happen. If you have an electron, pretty much who's coming along with it? A proton, all right? So you have this electron, but the proton's like, wait for me, buddy. All right? The protons are always kind of like following them around. And so technically what is actually moving is an actual hydrogen atom, right? It's an electron with a proton. If you have one electron, one proton, you are hydrogen. All right? You are hydrogen, all right? So here we have a disturbing muppet of some sort. but this is supposed to be some kind of organic molecule that has a couple of available hydrogens, all right? And if you have hydrogens that are available, which means that they're kind of easy to take, that means that you have available electrons, okay? So just keep that in mind. So in organic molecules, when redox reactions are happening, when electrons are moving around, you don't really see the electrons moving around, you see hydrogens moving around, okay? And it's whole hydrogens, not just protons. Protons don't have much energy. Hydrogens do, all right? Because they have electrons. Protons don't have electrons. They don't have a lot of energy of their own. All right? So, where do we take those electrons? Well, we have these special molecules that we've talked about before called electron carriers, like nicotinamide adenine dinucleotide, right? One of your B vitamins, which is a coenzyme, which means it completes the active site of certain redox enzymes, okay? And what it can do is it can easily bind to electrons, which is basically extra hydrogen atoms. And when it gets those extra hydrogen atoms, kind of split its eye apart, but that's what it looks like. It holds two electrons in one of the nuclei, but the other hydrogen nucleus is just kind of around somewhere. It's not actually bonded. So technically the way we write it, the accurate way to write it is NADH plus H plus, all right, when it gets reduced. Usually we just write NADH because we get lazy. All right, but in the back of your head, remember the proton. Always remember, okay? That proton's around somewhere. You don't have to worry about it now, but if you take some biochemistry, you will. Let's see, yeah. So basically, when you oxidize something, since you're removing electrons in the form of hydrogens, you're actually doing what we call a dehydrogenation reaction. And so if you're reducing something, it's getting hydrogenated. All right? So when you hydrogenate something, you give it more hydrogens, which means you give it more electrons, which means you give it more energy. Okay? All right? So here's your little reminder. So here's a simplified, they didn't even draw out the ribose because they were lazy. But here's NAD. Remember, it's got the this funky ring structure. It's a pyrimidine. It's got a positive charge on the end there. And let's see what else moves. And then this carbon, this double bond, can also be used to bind to a hydrogen too. So one of the electrons goes and binds to the nitrogen, neutralizes it, and breaks that double bond there. And the other one breaks this double bond, and you see the hydrogen kind of hanging out here. And there's a proton out in space somewhere. So there's an electron here, there's a whole hydrogen here, and a proton out in space if you want to be anal and balance the reaction completely. So there it is. Right, so this is reduced NADH, and this is oxidized NAD. Who has more energy? NADH. Okay. All right. So another electron carrier we're going to see a lot of, and we talked about this one before too, is FAD. Right, flavin, adenine dinucleotide. Right, flavin, riboflavin. There's all kinds of flavins out there. All right. It's got 3 rings. What did I tell you guys about these rings? Yeah. So they have these alternating double single bonds. It's called a conjugated bond system because the bonds are shared. The electrons just kind of flow around, makes it very stable. Same thing here. See the conjugated bonds here. There's some right here, right? Double bond, single bond, double bond. All right. So there's a little bit of conjugation right in there. That makes it very stable, so it can handle the extra energy of holding some extra electrons or extra hydrogens. Right. That's actually how. This is also how chlorophyll works. Chlorophyll is conjugated double bond systems all over the whole freaking molecule. That's why it can hold the freaking light energy of the sun. All right. So FAD picks up some electrons, but they also come with their buddies, the protons. You can go out and play, but you have to take your little brother with you. Okay. And then they reduce this nitrogen. See, you add a hydrogen to it. All right. And who else do we reduce? Oh, this nitrogen too. Right. So the two opposite-sided nitrogens get hydrogenated, and that's how you carry around those extra electrons. Right. As I said, so we're adding 2 electrons to each one of these. How much energy do you think 2 electrons carry? I mean, not quantitatively necessarily, but. Electrons have a lot more energy than like those phosphates we talked about before, like when you're putting phosphates on ATP. So FADH and NADH, these are way more energetic than ATP even, way more. Got it? And if you're more energetic, yeah, you're a lot less stable. Remember I told you guys that once you make ATP, you have to use it pretty quickly because it will just break down. It can only diffuse a little way across the cell. In a eukaryotic cell, it's not going to be very far. Right? You're going to use it within the same organelle or right near there or something. But these guys, they can't even move. Right? So these guys, they take the electrons, they can't even move. Remember, they're coenzymes. They're actually in the active site. So in the active site, they take the electrons from something and pass it to this guy because they're all in the active site together. Right? This is the coenzyme. Does that make sense? So there's not even time to diffuse. Because they're that high energy, that unstable. They're like, I got it, it's like a hot potato, right? I got it, here. All right, just passes it to the next molecule and reduces it. But it's crucial, as you guys will see. If we don't do this, we die. Very quickly. All right. Isn't this fun to talk about? Let's see how things work, then talk about how we die if they don't. Yes.
So they're passing the energy to the substrate?
Yes.
Yes.
Well, so the substrates might be in 2 parts. It'll be taking energy from one substrate and passing it to another.
Oh, I see.
Yeah, so it's doing the redox, right? It's always both. Right? So they take the electrons from one thing and pass them to something else. Right? Or pass them somewhere. All right? So, how do we make ATP? Well, there's actually 2 different ways that our cells normally make ATP, and we're going to go through both of them because they both happen in the same processes. So the main way, the general equation essentially of making ATP is you start with adenosine diphosphate. Do you remember why we have the squiggly here?
Energy.
Energy, how much energy?
A lot.
A lot, which means the bond is very
Unstable.
unstable, it's reactive, okay? So we have 2 phosphates. They already don't like being together very much, all right? These are like brothers that hate each other, okay? then the third brother, both of them hate, even though he's kind of friendly, all right? But to make them hang on to that third brother, you have to add even more energy. And the energy of adding the third one is way more than the energy to adding the second one, all right? So it's like a lot of energy to really make that stick on. But if you added a lot of energy, it also means that this bond is extra unstable, okay? And since we added energy, we input energy, it also means that this reaction is endergonic. Good job. And when we reverse it and we split it off again, it becomes, say it again? Exergonic, yeah. I heard a smattering of both things. All right? So here we have adenosine triphosphate, all right? And who's the least stable? The last phosphate. Did I tell you guys their names? They actually have names. Huey, Dewey, no. Yeah, so the first phosphate is the alpha phosphate, and this is the beta phosphate, and this is the gamma phosphate. Yeah, you don't have to know that for my purposes. It's just I like language. But if you're ever reading about ATP or something, you might see them say, well, the gamma phosphate or the alpha phosphate or something like that. They don't know what they're talking about. So if you want to think in anatomical terms, the proximal phosphate is the alpha, the medial phosphate is the gamma, or the beta, and the distal phosphate is the gamma. All right. So the first way that we will see that our cells make ATP, and guess what, everybody? All cells make ATP this way. Only some cells make ATP the second way. All right. But all cells make it this way, even if they make it the other way. I know it's confusing, but just follow me. So the first way we're going to make ATP, and the way that all cells make at least some of their ATP, is called substrate level phosphorylation. It's a very long and kind of vague sounding term for something pretty simple. All right. What that means is this is how you make ATP. So you, as usual, you start with ADP. And you're going to add a phosphate. But the phosphate is already attached to some kind of organic molecule, okay? So if an organic molecule passes the third phosphate to make ADP into ATP, it's an organic substrate passes the phosphate, then we call it substrate level phosphorylation, all right? The other way, an enzyme just grabs a sort of a free phosphate in solution, PO4 3 minus, right? You guys remember phosphate, PO4 3 minus. If an enzyme just grabs phosphate out of solution and smacks it onto ADP to make ATP, yeah, it's oxidative phosphorylation. It's going to use electron energy to do that. That's why it's called oxidative phosphorylation. So that's the second way. So, so all cells can do it this way, but only if you have oxidative respiration in your pathways can you do the other way. All right? Can you actually do oxidative phosphorylation? Okay. And so now the organic substrate has been dephosphorylated, and ADP was phosphorylated to make ATP. Okay. So now it has a lot more energy. Mm-hmm. Yes?
When does the inorganic phosphate come in?
That's later. Yeah. There's always little phosphates floating around. And they don't have by themselves, when they're not bonded to something, there's a lot of energy in this bond. If they're not bonded to something, the energy has to come from somewhere else. And spoiler alert, the energy is going to come from electron transport. All right. So the energy from electron transport is going to be used to basically take that kind of boring phosphate in solution and smack it onto to the ATP. Okay, that's where it comes from. All right. So, as I've been alluding to for the last week practically, when we start breaking down molecules, what we're really doing is we're pulling off their electrons, okay? And as we pull off electrons, we're taking their energy, okay? So when you oxidize something, you take its energy. You steal its soul. All right? And then you take that electron energy and you pass it to another compound, which becomes reduced, okay? And all this is going to happen in multiple steps. And eventually we're gonna do this process that has one of my favorite words in science. It's called chemiosmosis. Isn't that a pretty word? It's a very confusing word because it's hard to figure out what it actually means, but we'll come back to it later. But it's just have that as a word somewhere in the back of your brain. Shelve it for now. We'll come back to it. All right. The cool thing is, when our cells make most of our ATP through respiration, we strip electrons off, we put them through an electron transport chain, and that generates this huge buildup of energy that causes this big enzyme to basically rotate. Yeah, ATP synthase. So it makes this enzyme rotate. And when the enzyme rotates, it's like, I have phosphate, I have ADP, be as one, all right? And it just kind of slaps them together and you make ATP. So that's chemiosmosis. I'll explain why they use that word in a little bit. It's kind of confusing. I'll explain probably next time, actually. But chemiosmosis, or basically oxidative phosphorylation, which chemiosmosis is part of that, is also used in photosynthesis. So that pathway also exists in photosynthesis. So in photosynthesis, light hits these very efficient energy absorbing molecules called chlorophyll. That's what a pigment does. It absorbs light energy, right? If a pigment didn't absorb light energy, you wouldn't call it a pigment because it wouldn't have color. All right? If things don't absorb light, all the light bounces back and you just see white light. If it absorbs something, like for example, if a pigment absorbs red, you see the opposite color because the redness got removed. So what would you see? Do you guys know your color wheel?
Yeah.
Green, yeah. But you see the complementary color. If it absorbs purple, you see yellow, yeah. If it absorbs blue, orange. Orange. You guys don't know your color wheels. I was an art student, remember, so it all comes together. I remember I made this really fancy color wheel in one of my classes because we had to learn how to blend paints. And my roommate's like, you should submit that to the art contest. I'm like, yeah, my color wheel. Put it on display in the campus museum. Anyway, so light basically, it hits chlorophyll. And as I mentioned, chlorophyll has all these conjugated double bonds, right? And so the electrons bounce around, and they're so high energy that it bounces around for a little while, but then it eventually releases it. But the important thing is the pigment holds on to it long enough to release it to a particular receptor, all right? And there's a protein that takes the electron from chlorophyll and puts it into an electron transport chain. So the same process that happens in mitochondria is the same ending process that you have in chloroplasts. Pretty cool? So we'll just take a brief look at photosynthesis towards the end of the chapter. All right? So at the same time, we're gonna use electron transport to make ATP using the same enzyme, and some of you already knew it, ATP synthase. So chloroplasts also use ATP synthase. And this also is another type of oxidative phosphorylation. So oxidative, because we're using electron energy, right? We're taking electrons to do it. And phosphorylation, because we're taking phosphates and making ATP. All right, that term makes sense. The chemiosmosis is less sensical. All right, metabolic pathways. So we talked about metabolic pathways having multiple steps. So this is basically a very highly oversimplified of what we're going to look at for like the next week. All right. So you start with some beginning molecule, and this beginning molecule, this starting material here, which guess who this might be usually? Glucose. Yeah, it's glucose. It has some pretty energetic electrons, and remember, energetic electrons are always carried in the form of hydrogens. So we're going to take some hydrogens away from glucose, and that's going to help us make NADH. All right. Then you make an oxidized product, but there's still some energetic hydrogens there. Glucose is just full of hydrogens. All right. Fats and sugars have hydrogens all over the place. All right. So your cell's just like yes. Okay. They just want it. Rearranges some bonds and such, and actually you can release enough energy to also make some ATP on the side. This is substrate level phosphorylation, as we'll see later. So some substrate level phosphorylation happens later, but ultimately we collect all these electrons, And the electrons go down this chain, and as they move, as the electrons move, they're actually releasing energy. So they get passed from carrier to carrier to carrier to carrier. I don't remember how many there are, there's a lot. But I mean, there's three big complexes, but there's a couple in between carriers, but each one has a subcarrier inside of it. So even one big electron carrier protein has multiple carriers inside. So the electrons get passed multiple times, and each handoff releases a little bit of energy. Little poom, poom, poom. All right. So it's not quite the snap. All right. Of that infinity gauntlet, but you're just doing little sparks, like. All right. And so the molecules get rearranged until they're as many electrons as you can easily strip off are stripped off, and the last acceptor for those electrons is oxygen. And since electrons are being passed in the form of, when oxygen accepts a bunch of hydrogens, it tends to form water, okay? And because we're breaking down carbon compounds, we're stripping off hydrogens, but we're also, at the end, we're eventually going to break all the carbon-carbon bonds too. And when carbon gets completely oxidized, it turns into CO2, right? So that's our end product. That means once you have all the carbon dioxide at the end, it means you've taken off all the hydrogens and any possibly available electrons, okay? So you've taken all the electrons off that you possibly could, and all those electrons are going to be used to make as much ATP as we can, and as you guys will see, it's a lot. All right, so we're going to start with carbohydrates, then I'll give you guys a little side overview at the end of how we also use proteins and fats for energy. The short version is we basically break the functional groups we don't need off the fats and proteins, and we turn them into carbohydrates and put them in the same pathway. That's pretty much what happens. You just make them little simple carbon backbones and put them into the same pathway. So there are 4 steps. You might usually hear about 3 steps, but we're going to do all 4 because we aren't wimps here. All right, because it doesn't actually make sense unless you look at all 4. So we're going to start with a process called glycolysis. Who's heard of glycolysis before? All right. Have you ever thought about what the word actually means? Because what it means is what it actually does. Yay. That's not always the truth, but in this case it is. All right. Glucose, yeah, sugar. Lysis. What are we going to do to the sugar? Break it. Not just break it. Remember what lysis actually means? Split. We are literally going to split it in half. All right. How big is glucose? 6 carbons. Guess where we're going to break it? Between 3 and 4. Right? Yeah, so between 3 and 4, right? So if you break between carbon 3 and 4, you have 1 through 3 and 4 through 6. Okay, that's what we're going to do. Okay? So you break it in half, and that's not the only thing. We split it, that's the first step, and then we start stripping off some hydrogens too. Okay? And that's going to release a lot of energy. So we're going to make some ATP here, and then if you have respiration, okay? So you guys you're probably used to hearing this term cellular respiration, okay? There's cellular respiration, and then there's also like bodily respiration, right? Which more properly, what you should call that is ventilation, right? So ventilation is inhaling and exhaling, okay? Cellular respiration, when you guys hear that word, it's such an old term that to help you think about what it actually is, if you hear cellular respiration or just respiration in reference to cells or microbes, automatically think electron transport chain, because that's really what it means, okay? If you don't have an electron transport chain, if you don't have oxidative phosphorylation, which is coupled to the electron transport chain, there's that word, coupled. Uh-oh. You know what's coming. If you don't have electron transport and oxidative phosphorylation, you don't have cellular respiration. Okay? You have something else. All right, which is usually fermentation. All right. So if you're a fermenter, you basically stop here. Remember I said fermentation is just glycolysis with a couple finishing steps, essentially. All right. But if you have a respiratory pathway, then you're going to keep oxidizing even what you've already oxidized, because if you have respiration, you can fully oxidize organic molecules, and if you can fully oxidize them, you can take all of their available electrons and energy. Yeah. So the end product of glycolysis is a compound called pyruvate. So this is a one-step pathway. This one right here, that's the easy one, where we're just going to oxidize pyruvate. But when we oxidize pyruvate, what it does is it gets it ready to enter the Krebs cycle. Okay. So we're going to take electrons here, and even those electrons go into our electron transport chain. Right. We're going to take some electrons here. Those go into our electron transport chain. Electrons here go into our electron transport chain. But where we get the most electrons is right here. The Krebs cycle, also called the TCA cycle, also called the citric acid cycle. Those are all accurate names. I'll explain it all to you at some point. All right. The Krebs cycle, because it was discovered by Dr. Krebs. All right. The other two are the actual proper biochemical names for the process. And then finally, so these three processes harvest lots and lots of electrons. All right. And all those electrons are going to be carried by, do you remember who? NADH and FADH2. That one actually carries two hydrogens. Right? FADH2. And NADH and FADH2 are going to put their electrons into the electron transport chain, and that is what is going to lead to oxidative phosphorylation, making ATP. Right? So that's your quick summary. Don't worry, I'll present it lots of different ways. So here's the incredibly oversimplified version of glycolysis. So in glycolysis, we start with our good buddy, glucose, and we're going to split it in half and steal a few electrons too. See this guy? And there's not just one. It's going to make at least, two. Thank you. You've already been studying. Look at you. That's what you all should have read this chapter already, right? That's how it works. It's on the syllabus to be read like a week ago. So keep up with your reading. So when you split glucose, you're going to take some hydrogens. That's to release a lot of energy. And some of that energy is going to be used to make ATP. But we're not doing oxidative phosphorylation yet, so this must be from substrate level phosphorylation. So you're already looking at that word. And eventually we're going to end up with a partially oxidized product called pyruvic acid. And if you don't have a strong oxidizer around, what's our favorite strong oxidizer? Oxygen. Okay? If you don't have a strong oxidizer around, you're done here. Right? You do a couple steps to keep this from building up too much, but other than that, you're kind of done. All right? But if you are a respiratory organism, then you keep moving on. All right? So let's look at glycolysis in more detail, because I know you guys are going to love this. You ready? Now this is the simplified form, and you don't even have to know every step. So as we learn the biochemical pathways, everyone, I want you to pay attention to two things. I told you this before. You know what the two things are? Well, maybe I didn't mention them yet. There's two things I want you to pay attention to. Is when we start a pathway, how much energy goes in, and in what form is that energy? And at the end, how much energy comes out, and in what form? Okay? Energy in, energy out. All right? That's one set of things. The other set of things is how many carbons go in, how many carbons come out, and in what form. All right? So you don't have to know every individual step. You just have to know what goes into step 1 and what goes into step Z, or comes out of step Z, or whatever the end step is. All right? So when you keep stuff from your kids' toddler days, you can find uses for it. So I got my pop beads that my kids used to play with. All right? Here's glucose. How many carbons? 6. All right? Here's what our cells do. When, let's take just for the fun of it, let's take a eukaryotic point of view. All right? In eukaryotes, glycolysis happens in the cytoplasm. See? Got a reader here. Glycolysis happens in the cytoplasm. When it's in the cytoplasm, it gets it gets bound by an enzyme. Let's put it that way. My brain's too scattered right now to use the right word. So it gets bound by an enzyme, and the enzyme's going to stick a phosphate at one end. It's actually going to attach it to carbon 6. Okay. Then a few of the bonds are going to be rearranged on the molecule, and it's going to make another carbon available, carbon number 1. And we're going to put a phosphate there. all right? So we already put one phosphate on. What does that do to the molecule? Makes it more energetic. Now we have a phosphate at the other end, all right? These phosphates are only 6 carbons apart. How much do the phosphates like being close to each other? Not. What does it do to the molecule? Yeah, it's super unstable. Guess what it's going to do? It's going to break right in the middle. Okay? So you see, so we actually use ATP to phosphorylate carbon 6, all right? And then we're going to use ATP again, rearrange a few things, and use ATP to phosphorylate carbon 1, all right? This is glucose 1,6. It's called bisphosphate, all right? You use it, so remember we had diphosphate for ADP. That's because the phosphates were connected to each other or they're on the same carbon. If they're on separate carbons, it's called bis. That means there's 2, but they're separate. Just a fun fact for you. You don't have to memorize it or anything. Just to tell you why I use that term, and your book does it because they aren't biochemists apparently. but this is fructose 1,6 bisphosphate. Okay. And if you go into your textbook and go into the appendix, it will actually show you the atomic detail of each of these molecules. But I don't want you to be distracted with that. Okay. I just want you to know what's coming in and what's going to go out. And so when we add the 2 phosphates, the bond in the middle becomes very unstable and it breaks. All right? And you end up with these 2 3-carbon phosphates down here. But we just broke a bond, and so that bond is going to release a whole lot of energy. All right? This is a huge energy release. This first breakage is a huge, huge energy release. All right? So 2 ATPs are used. All right? So in order to make ATP, we have to spend some ATP. Yeah. If you're going to make money at the bank, you have to put in some money, right? All right. So these 2 3-carbon molecules, it's called glyceraldehyde 3-phosphate. All right, you don't have to worry about that too much, but that's what it's called. All right. So here's glucose 6-phosphate. All right, and then it rearranges and actually rearranges into a fructose. Remember, fructose has carbon 1 and carbon 6 sticking up. And so now carbon 1 is available, and we phosphorylate that. And now we have fructose 1,6 bisphosphate. All right. And one of my favorite terms in biochemistry, just for fun, this enzyme at step 3 here is called phosphofructokinase. I just like to say that. Phosphofructokinase. Because it's going to take fructose phosphate and put another phosphate. You guys know what kinases are? They're enzymes that add phosphates to things. You probably learned about some of these in physiology.
So if fructose is the initial product, it takes less energy to break down.
What takes less energy?
Because if we start with fructose, then we don't need the ATP from step 1.
Yeah, if you start with fructose, it gets phosphorylated and jumps right into here. But it's still going to have to be phosphorylated. So you're still going to have to spend an ATP. It's just a sidestep. Yeah, there's other enzymes that sidestep other sugars in. So all other sugars will feed into this pathway somewhere, just so you guys know. Because remember, most of the other sugars that we digest are also 6-carbon sugars, and they just get rearranged until they basically look like glucose or an intermediate of glucose. That's pretty much what happens. All right? So it breaks into two 3-carbon things. Glyceraldehyde 3-phosphate and also dihydroxyacetone phosphate. But this one, you can just ignore this one because it just gets rearranged into a second molecule of this. So we just have 2 glyceraldehyde 3-phosphates essentially. So we had one 6-carbon compound, now we have 2 3-carbon compounds. And the important thing to remember here is that now when we're looking at what happens and we're doing the calculations of how much ATP is going in and how much ATP is coming out, everything happens
In pairs.
twice. Yeah, it happens twice. All right? And just because I know you guys love your structures, so here's fructose 1,6, see? 1,6, 1 and 6 bisphosphate, and it gets rearranged, it gets isomerized, and you get glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. But this one will rearrange and become that. See, the hydrogen just moves. That's all it does. I know a lot of you like biochemistry, so there you go. Yes, I'm being sarcastic. All right. So that was what we call the energy investing stage, because we just had to spend 2 ATP to get things going, okay? And then we had that big split, right? We made those 2 glyceraldehyde 3-phosphates, And so that big split between the carbon atoms, that released a whole lot of energy. Let me show you how much energy it released. It released so much energy that those electrons were used to make 2 NADH. Okay? We haven't talked about numerical things in a while, right? You ready? I know you guys love numbers, right? We do some math. All right. Let me just quantify a few things for you. ATP, the amount of ATP, amount of energy in an ATP molecule is about 12 kilocalories. What does that mean, kilocalories? 1,000 calories per mole. Okay? There's 12 kilocalories per mole of ATP. That's a lot of energy, just so you all know. Okay? When we make NADH, and if you want to be extra accurate, you can do that. When we make NADH carries about 52 kilocalories per mole. All right? So there's over 4 times as much energy in NADH as there is even in our buddy ATP. All right, the golden child over here. All right? So gold coins, platinum coins. All right? And then we have FADH2. FADH2 is sort of the little brother to NADH. And FADH2 has about, let's see if I can remember now. I'll have it on one of my slides later, but I think it's 43, something like that, kilocalories per mole. I still got summer brain. Something like that, it's in the low 40s. 43 kilocalories per mole, right? So do you guys see how much energy these guys are carrying? And that's why they are highly, highly unstable. They can only hold those electrons for a moment. They can't even leave the active side of the enzyme, right? They just hold it and pass it. That's all they can do, you know? I don't even know how fast that is. It's probably like a billionth of a second or something ridiculous, all right? But look, we made 2 NADH, so that should tell you that is a buttload of energy that got captured right here. Got it? All right? Not only that, but not only did we make 2 NADH, but there was so much energy released that we're actually able to add 2 phosphates, 2 high energy phosphates, 1 to each of the glyceraldehyde 3 phosphates. So now, each of our, remember this is happening twice, we're just going to hold one until we have one hand available. So each of our phosphates, so the oranges are the phosphates, right? Each of these 3 carbon compounds now is going to have 2 phosphates. And now we have 2 phosphates that are only 3 carbons apart. There's a lot of energy there, okay? And notice, how did they draw that last phosphate? It's not a good pointer. So see, it's got a high energy bond. You guys see that? It's a high energy unstable bond, okay? That means that these phosphates don't really want to stick around, okay? But guess who's nearby? So we have some high energy phosphates, and ADP comes wandering by, minding its own business. All of a sudden the phosphates are like, yeah, and they like ambush it. Because remember, the 2 brothers don't really want the 3rd brother to come along, right? But he just kind of like ambushes them, jumps on, and we make 2 ADP. So 2 phosphates from here attach to 2 ADP and make 2 ATP. So we've made our first 2 ATP, everybody. So we have our first 2 phosphorylations, or ATP phosphorylations. And what kind of phosphorylation was this? It came from an organic carrier, so it's substrate level phosphorylation. An organic molecule donated the phosphate. All right? Then that leaves us, so that was 1,3-bisphosphoglycerate. You don't have to know the names, I'm just pointing them out because it's the way I think. And then we're left with 2 single phosphate molecules again, 3 phosphoglyceric acids. And what happens is, and they're not going to show you here, but what happens is the phosphate on carbon 3 here, it gets moved to carbon 2. So now it's 2 phosphoglycerate. And then a water molecule is removed. So we remove an oxygen and a couple hydrogens from here. And what that does, they're not showing you, but just put this in your head. It makes a double bond right here. So there's a double bond right here on this carbon, and this carbon is trying to hold on to a phosphate. That makes this situation highly
Volatile.
unstable, right? See how so it gets energized there? So this carbon is trying to hold on to a double bond and a phosphate. All right, it's like I can only do so much, people. All right, and so how long can it hold on to that phosphate? Not very long, you know. And a couple ADP come floating by, and this phosphate's like, I'm jumping ship, and we make 2 more ATP by the process of substrate level phosphorylation. Okay, so how many ATP did we spend on the first half, the first 5 reactions? 2. How many do we make now? 4. 2 net, yeah. So if you went to the bank and you gave them $200 and you came back a couple weeks later and you got $400 back, would you be happy? Not a bad investment, right? Doubling your investment. All right? And then after this final phosphorylation or dephosphorylation of this molecule, the phosphoglycerate, you end up with 2 pyruvic acids. Okay? And then that's when we're getting ready to go into respiration, if the cell can do respiration. Okay? So we made 4 ATP produced for a net total of 2, right? But don't forget, we also made 2 NADH right here, right at the midpoint, at the big split. We shall call this the big glycolytic split. Okay. And so this is what I was going to show you. So this is 2-phosphoglycerate, it's phosphoenol pyruvic acid. So when it's got the double bond, it's an enol. And so you see it's got a double bond on the carbon and the phosphate, so that's not stable. What's the mistake they made in this diagram? Yeah, the phosphate should be negatively charged. Yeah. So even internet people are derps. All right. So glycolysis, what did we do? We took glucose, we added 2 ATP, 1 to each end of the glucose, and that made it very unstable. Okay. We brought in 2 ADP to accept and 2 naked phosphates, basically, inorganic phosphates because they're not attached to anything, and they came along, and the 2 ATP phosphates and the 2 free phosphates were added to ADPs to make a total of 4 ATP. We also harvested electrons in the form of 2 NADHs, all right? And so we doubled our return, and we have some extra energy, which is only useful if you have an electron transport chain, all right? Otherwise, these guys will just get recycled, all right? That's what fermentation does, by the way, everybody. That's the main job of fermentation, is to recycle your NADH back to NAD. You have to regenerate that coenzyme, otherwise you can't keep glycolysis going. All right? So far we've only made ATP by 2 steps of substrate level phosphorylation, right? So two steps, but 4 total ATP were made. Let's see. Yeah, hopefully this website still works, I like this one a lot. So this one's a nice little summary of glycolysis in video form. Yes, I am. What? Our computer's talking to me now. All right. I don't know why I can't remember myself. All right, here we go. So we're going to start glycolysis. You ready? First, we're going to spend 2 ATP. They're already waiting, right? So here's glucose, glucose 6-phosphate, fructose 1,6 bisphosphate. Right? Even they got it wrong. It's bis, people. All right? And then what's going to happen? It's going to split. All right? They're skipping a lot of steps here, but well, no, actually, they did it right. So it splits, and now we have 2 phosphates available, and a lot of energy was released, and we're going to use that energy to make 2 NADH, and also to add 2 phosphates. Right? Now all 4 phosphates will be harvested to make 2 ATP, and then 2 more ATP for a total of quotation mark. Right, we got 4 ATP. Right? So that's your products of glycolysis. 2 pyruvates, 2 NADH, and 4 ATP for a net of 2. Good job. And now we're back at slide 1 somehow. I'm not trying to pass that. Where were we? There we go. All right. Now where did I put my remote this time? I don't know how I always set it somewhere. Is it over here? ¿Dónde está el remote? Oh, all right. So that's glycolysis, and a lot of cells can survive on glycolysis. Animal cells cannot. All right. We're way too high maintenance. Tell your friends. Eukaryotes, especially animals, are very, very high maintenance. All right. And so what we're going to do is we had glucose and we oxidized it until all we had left was 2 pyruvates. So pyruvates are already partially oxidized. So we've taken some of their high energy electrons, the easiest, the low-hanging fruit. All right? We took the low-hanging fruit, the highest energy electrons, the ones that are easiest to remove because they're the most unstable, and therefore have the most energy. But now, if we want to fully oxidize it and really get all of that energy out of that tank of gasoline, all right, we have to do oxidative metabolism. So we're going to oxidize the pyruvate even more and harvest even more electrons to give even more fuel to our electron transport chain. Yeah, ETC. Did my remote go? Anyone see it? I have no idea where it is. Oh, it's under my glucose molecule. All right. So we're going to make lots more ATP by our good buddy, oxidative phosphorylation, and it's a very good buddy to us because we die without it, like within minutes. So I'll just introduce you to our transition step. Your book calls it the intermediate step. That's lame. I call it what it is, pyruvate oxidation. We're going to take our pyruvic acid and we're going to take some of its hydrogens, all right? But in the process of taking its hydrogens, we're actually going to split off for the first time a carbon. And whenever we split off carbons, we split them off in the form of CO2. So glucose, we started with how many carbons? 6, all right? We had 6 carbons, then we split them, and we ended up with 2 pyruvates. So how many carbons do we have left? We still have 6. We haven't lost any yet. But now we have 2 3-carbon pyruvates, and we're going to split off for each pyruvic acid. And how many pyruvic acids do we have?
2.
We're going to split off a carbon dioxide. So we're going to lose 2 of our carbons. How many glucose carbons do we have left? 4. All right. And when we split them off, we're going to take the electrons and make 2 NADH, but we're also going to make 2 acetyl-CoAs. CoA is this big, complicated organic coenzyme. I'll show it to you later, but it's kind of scary. It's like really complicated. But the main thing is it's very good at carrying 2 carbon groups, which we call acetyl groups. And the acetyl group is what is going to enter into the citric acid cycle, the Krebs cycle. And then we're going to really get down to business, stripping off all the electrons possible and really powering up our electron transport chain. And the more you power up your electron transport chain, the more you can make ATP. Okay? So remember, this step happens twice for every glucose that went in. Right? Twice per glucose. Oh yeah, I guess I didn't put it down here. That's coenzyme A. Isn't it beautiful? Look, it's adenine. Yeah, it's adenine. So it's actually a nucleotide. And then it's got a couple phosphates and this weird organic linker with nitrogens in it. And the acetyl group is attached to a sulfur. This is a high energy bond. Remember that. That high energy bond is going to allow us to pass it into the Krebs cycle.